Thank you so much. So first of all, thank you so much for the invitation to present here in the introduction to present my works. And well, long story short, what I've done during my postdoc is to come up with a new way of making mouse brain organoids. And of course, when you think about the mouse, like the idea of mouse brain organoids, the first question that always jumps into your head is to what do we need that for? Why do we need a mouse brain organoid when we already have human brain organoids and we already have mouse in vivo? So there's like a variety of reasons why a mouse organoid system could be useful in different fields. But for example, and I will describe some later on, but for example, if we think about cross-species comparisons, we want to compare human versus mouse. If we use human organoids in vitro system versus mouse in vivo, it's always complicated to discern what is an in vitro artifact from what is genuinely a cross-species difference. So for example, in this paradigm, I do believe that having a mouse organoid system can help us bridge the gap between the in vitro and the in vivo systems. And of course, we're not the first ones to think about making mouse brain organoids. And early in the early year 2000s, Yoshiki Sasai, the team of Yoshiki Sasai were pioneers actually in the field of developing first mouse brain organs and then human brain organoids. And even though his works were pioneer and really awesome, nowadays they do lack reproducibility. They are not as robust as human brain organoids. And we do believe that, well, there's a variety of reasons why they are not robust as the human ones, but I do think that the main reason is because of the starting cell type. So when it comes to mouse stem cells, as probably you already know, most people tend to work with ESCs, so mouse ESCs that represent the naive stage of pluripotency. Whereas when it comes to human stem cells, human PSCs, people tend to work with the prime state of pluripotency, being the prime state, the state of pluripotency that is ready to be committed into ectoderm, endoderm, and mesoderm. And I do believe because of Sasai's protocols starting from ESCs, that was adding a major bias in his protocols. So indeed, one of the first things that I did during my postdoc is to optimize the way that we culture mouse stem cells, not in the naive, but in the prime state. So I optimized the way that we culture mouse epiblast stem cells, or EPSCs, thinking that this will be a much more suitable starting cell type to build better mouse organoid systems. And indeed, that's what I did during my postdoc. So starting from mouse epiblast stem cells, we subject them to a variety of cocktail molecules to try to recreate brain development in a dish. And well, before going any further and showing you what we get when we run this protocol, just let me give you a brief overview about how do we develop a brain, how do we develop a cortex more specifically. So in an oversimplified manner, when it comes to cortical development, most of the cells originate from the cortical progenitors or radial glial cells, RGCs. And what will happen is that over time, these RGCs will start giving rise first to preplate neurons, then to deep layer neurons, then upper layer neurons. And as time progresses during late development, these progenitors, these RGCs, will become less neurogenic and will start to become gliogenic and will start giving rise to astrocytes and oligodendrocytes. So now the question is like, am I able with my protocol to reproduce this cell diversity that happens in vivo? So now, yes, some images. So here I'm showing you an early stage mouse organoid. Importantly, we can see that they do express the canonical cortical marker FOXG1. And as hopefully you can appreciate here, already by day 6, we have a huge number of cells that express CTIP2. CTIP2 is a marker of deep layer neurons. So then after seeing this, I dig a little bit further at later stages into whether we can actually recreate all of this neuronal diversity that we found in the mouse cortex. And indeed, I could find at later stages that we do have both. We have deep layer neurons characterized by the expression of CTIP2 and TBR1, and we do also generate upper layer neurons characterized by the expression of CAX1 and SATP2. So the next question was like, so we can generate the neuronal repertoire that exists in vivo. Can we also recreate what happens with those progenitors that at some point stop giving rise to neurons and start giving rise to astrocytes and oligodendrocytes? And indeed we do. So as you can see here by day 16, we do have already a significant number of GFAP positive astrocytes, and the same for the oligodendrocytes, here labeled with OLIG2, showing altogether that we do seem to be able to recreate the cell type diversity existing in the cortex, or most of the cell type diversity. And the final validation that I did to be sure that what we're recreating somehow resembles the in vivo cortex, I did single cell RNA-seq on day 12 mouse cortical organoids. and I compare it to an in vivo data set generated by the Arlota lab. As hopefully you can appreciate here first in this graph at the left with the integration of both data sets, and here subdivided by whether if it comes from the organoid or from in vivo, what I found is that our organoids do indeed have a close, like a high degree of resemblance to their in vivo counterparts, showing altogether that with this protocol that I came up with, we can recreate the cell type diversity of the developing mouse cortex, and we can do so in a timely manner following in vivo kinetics of the mice. And well, so the next thing that we check is like actually one of the benefits of recreating mouse in vivo kinetics is that we could also potentially recreate postnatal traits much faster than in human organoids. Human organoids to recreate postnatal things might take months to years to do so. Mouse, if we are really following in vivo kinetics, should do that in a matter of weeks. And indeed we do. So for example, about the different things that we check, I look into neuronal maturation. So as you can see here, in a matter of few weeks, we do see an accumulation of MCA methylation. MCA methylation is a hallmark of postnatal neuronal maturation, and hence this increase in methylation shows that we're getting postnatal mature neurons in our organoid system. And the same is true for all of the cell types that we look at. Here I'm showing you some oligodendrocytes at the right, stained for MVP, where you can see that these oligodendrocytes get really complex with the ramifications over time. And we've actually seen that these oligodendrocytes are capable of myelinating the surrounding neurons. So great, also we have a system that recreates mouse brain development. We can use it for cross-species comparison, but what else? So actually there's this great opportunity of working with different mouse systems. That is the huge diversity that exists of mouse subspecies. Different mouse subspecies are a great reservoir of genetic diversity. So just to give you some numbers, if we are to compare the strain Black 6 versus the subspecies Spretus, we'll find that they are different for about 50 million polymorphism or SNPs. Whereas if we are to compare to humans, to random humans, we'll find between 4 to 5 million polymorphisms only that set them apart. So a huge diversity across mouse subspecies that does reflect in major changes in neuroanatomy. So it reflects in the way that they develop their brains. So you can see here at the left, comparing the brains of a Black 6 with a Castaneous, you can see that there's an obvious change in the size and even in the size of more specific structures. And even if we get to the more fine-tuned neuroanatomy, when we compare the hippocampus of the Black 6 and the Spretus, you can see that there are major changes in there. And not only do they have different neuroanatomical features, but it's well known that different mouse subspecies have differential disease susceptibility. So then the question remains, how does this huge degree of genetic diversity impact so much development as to affect neuroanatomy and disease susceptibility? And here's when the great thing of working with mice comes, is that we can breed them. So since we can breed those different subspecies, we can generate F1 hybrids to study how changes in gene expression across subspecies may affect the way that they form the brains. What is great about working with an F1 hybrid is that within a single cell, we have both alleles. We have the Black 6, and in this case, the Spretus. So we have a completely controlled environment that is not biased by cell type, batch, or whatsoever, where we can efficiently study how a gene gets expressed and when from the Black 6 allele or from the spread allele. So the idea here will be to look for genes that are differentially expressed between the spread and the Black 6 allele over the course of development, and trying to find those that might explain the differential neuroanatomical features that we can find in the mice. And here, the only downside that exists is that crossing these mice, Spretus and Black 6, can be a nightmare. It can take months to years. And if you want to do like a time lapse of brain development, it can become many years of work. So we're wondering why not to use organoids for this. We only need one cell and we can do organoids and brains as much as we want to study this trait. So indeed, that's what I did. So I did not only work with one F1 hybrid between spread and Black 6, but I had 3 other wild-derived strains crossed with Black 6, Castaneous, PLDK, and MOLF, extract the fibroblasts, convert them into epiblast stem cells, and then generate cortical organoids out of these 4 different highly divergent mouse backgrounds. And among many things that we did, one of the things that we did is to actually see what we have within those organoids. So here I'm showing you single-cell RNA-seq analysis on day 12 organoids from the 4 different F1 backgrounds. And as hopefully you can appreciate, across the 4 different F1s, we do get the same cell types. And this is really important for us because it's not only for the downstream thing that we want to do, but because it shows that our protocol is robust enough as to work across highly divergent backgrounds. And so far, we've seen that it works with more than 20 different cell lines from highly divergent genetic backgrounds of the mice. That it's quite important for us. So good. So we have this system, we have these data sets. Can we use it to analyze how they express different genes between the black 6 and the wild-derived strain over the course of development to justify those changes in neuroanatomy? So I won't go through all of the data. I will just give you like a sneak peek into what we found. And here it is. So basically, when we look at genes that are differentially expressed from the black 6 or the spread allele within the F1 over the course of organoid generation, so genes that are allelically imbalanced, we found that there are thousands of genes that are differentially expressed between those two strains. And what was even more surprising is that, as hopefully you can appreciate in this Venn diagram, a lot of these imbalances or changes in gene expression had a strong cell type specific component, suggesting that cell type specific cis regulatory elements are differentially regulated between black 6 and spread, and may be differentially regulating the way that they do express genes during the course of development. And this is important because it seems to affect genes that are well known to play a major role during brain development, such as ARID1B, SMARCA5, or even if we look at the astrocytes, the canonical markers of astrocytes, GFAP and aquaporin 4, are differentially expressed between those subspecies over the course of development. But then the only downside is that there's too many genes. There's like thousands and thousands of genes. So how do we go to the gene or the few genes that are actually changing the way that they form their brains? So as a starting point, what I did is that I crossed our database of thousands of genes differentially expressed with genes that have been previously associated or have been previously shown that when they are knocked out, they lead to an neuroanatomical change in the mice. So it's a data set of 200 genes that has been published. And as you can see here in this white part of the histograms, I could find dozens of genes that are associated with neuroanatomical changes that are differentially expressed between spread and black 6, suggesting that those may be underlying those changes in neuroanatomy. And as I mentioned before, another interesting thing is that different mouse have different disease susceptibilities. So how can we explain that? So I cross-reference our data set of imbalanced genes with genes that have been strongly associated with autism spectrum disorder, ASD. And once again, I found dozens of genes important for ASD progression that seem to be differentially regulated across subspecies. And indeed, they might be able to explain why different mouse subspecies have different disease susceptibility. So altogether, hopefully I've convinced you that I come up with a new system that can facilitate performing cross-species comparisons, that already within the mouse system itself, we can take advantage of the great genetic diversity tools that mouse provide us with to get insights into how complex traits are regulated by genetic variation. And yet another bright side of our system, as I said, is that we get cells to be mature in a matter of weeks, whereas in human systems, it might take years. So maybe if you want to study what genes are important for neurodegeneration or chemical screens, it might be potentially wiser to start with a mouse system, do a screening there, and then validate the hints in a human system. And of course, last thing to mention, I mean, we developed these protocols because we want to use them, but it would be great if they could also be used by the community. And that's our main goal, for people to adopt these systems and them to become something useful for them. So for that, we have a bioarchive that was published last year that comes together with a supplementary PDF. So you can have like a step-by-step protocol on how to generate these organoids. And please, I'm more than happy to discuss these organoids, how to adapt them into a lab. Any question, let me happy to help. And last thing, of course, no one does nothing alone. And I just want to thank you, the main people that were involved in this project, our collaborators from Australia, Emily and Veronica, that were pivotal for the allelic imbalance analysis, Thomas Verbuchen, that has directed this work, Tarek for his hands-on help, the funding agencies, and of course, I should mention that indeed I'm in the job market nowadays. So if you believe that these and other similar systems may be of use to your institute, I'll be more than happy to discuss with you. And well, thank you for listening.
Great. Thank you so much for the beautiful work. Yeah, we can start with Alice's question. So your organoids seem to have rosette-like structures within them. How variable is this organization, and is it important?
So the thing is, like, they start as a single rosette. Mostly they start like a single, like, neuroepithelia. But as they grow, I think that is not well sustained over time to have a single huge neuroepithelia. So they start to break down into smaller ones. So the starting point is the same one. What they become after, when they become so huge, I think that they need to break down into smaller pieces for physics reasons that I do not fully understand.
I see. Yeah, second question from Alex. Are there behavioral differences between the different mice strains?
Yeah, actually, yes, it's kind of cool. Like even like the lifespan is different. The spread are super jumpy, small, run everywhere. The black six there seem to be like aggressive but less active. So there are like major behavioral difference between them. So of course, like all of
I,
these changes in their anatomy, expression, everything, it translates into major changes in behavior as well. That makes them even cooler.
I see, I see. That's very cool. And we have a question from Etienne. Super interesting system. How does necrosis compare to your human organoids?
The crosses.
Um,
Crosses. Yeah, I actually that's a bright side of our system as well, because the organoids remain smaller. So our organoids are much, much smaller than the human ones. So there's no major necrosis that we can observe. I did a Cliff Caspace 3 time course over different days to see if at any point we have a major cell death. That does not seem to be the case. And I think that in humans it happens because they get so big that the internal part cannot really get oxygen from the outside. So yet another advantage of our system is that it doesn't seem to be a major cell death.
I see, I see. Um, I have another quick question. I remember there's from Lawrence Starker's group published like a combination cocktail that can like accelerate like differentiate neuron. I wonder, have you tried those in the organoids? And would they change the timing? And if the timing are changed, will the cell types generated be changed?
Yeah, that's a pity because they leave abstracts to us, literally, Lawrence Studer. Like they have their lab abstracts. Yeah, it's the gin tonic cocktail. I
Yeah,
think that would be interesting to try, but my assumption would be that if you force the progenitors to differentiate and mature sooner, you might be losing some cell types. You know, like you might not generate upper layer neurons if you have deep layer neurons that you force them to differentiate right away.
right. So I was curious if you like force like say human neural organoid differentiate a little bit faster, will you generate cell types more like the mouse brain organization?
Yeah, I think so. And I bet that they're working on that.
Oh, great. Okay.
Yeah, at the end, it's an artifact, not to force for them to mature faster. With the mouse, you don't need to do that because they will naturally mature
Yeah.
following the right steps, hopefully. But I saw that potentially those kind of combination of cocktails may help also the human system.
Okay.
Totally.
Cool. All right. So thank you again for your beautiful talk.